So we will be looking at the role of touch. Especially, we will be discussing the neural machinery that is involved in the perception of touch. And then we'll be talking about how the sensation of touch is getting transmitted all the way up to the brain. So if you look at the touch is actually an important type of somatic sensation. And there are several somatic sensations like pain, temperature sensations, the ability to sense body balance. And with regards to touch, the somatic sensation of touch begins at the skin. Okay, now skin is a very important organ. And in terms of touch, we need to consider skin as two regions. Now, if you look at it, there is a hairy region and there is a glabrous region, which is basically the hairless region. So if you can consider your palm, the backside of your palm is the hairy region, the palm itself is the hairless region, which is the glabrous region. Further, the skin is also divided into the epidermis and the dermis. Now, importantly, the skin is a very important protective organ. It's the biggest sensory organ. It helps in preventing the evaporation of body fluids. And then it also provides us a direct contact with the world. So, right, anything that we are encountering, the first point of contact there is the skin. Whether any type of insults, all the direct contact happens on the skin. Now, with regards to touch, the sensation of touch is actually taken forward by a set of receptors which are called as the mechanoreceptors. So the somatic system that works with relation to the touch are the mechanoreceptors. Now, when you just look at how critical the skin is with relation to touch, you just have to read these two lines and you will understand how important the role of the skin is. So skin is sensitive enough that a raised dot that is measuring only about 0.006 millimeter in height and 0.04 millimeter in width can be felt just by a stroke of our fingertip. So just compare that with the braille dot, which is used by people lacking vision to read. And you can see that the braille dot itself is 167 times higher. So you can imagine how sensitive our fingertips are with relation to the perception of touch. Now, when you talk about the mechanoreceptors on the skin and the mechanoreceptors that contribute to the sensation of touch, there are different types of mechanoreceptors. And these mechanoreceptors have been differently labeled, have been given different names. For example, there is a Pacinian corpuscle, there is a Ruffini's endings, there are the Meissner corpuscles, the Merkel's disc. Now, all these names come from the histologists who first identified them. Now, if you look at the organization of these mechanoreceptors and you look at the structure of the skin, you have on one side the hairy skin, the other side you have the glabrous skin. And then you can also see that the separation of epidermis and the dermis. You can see that the mechanoreceptors are distributed both in the epidermis as well as in the dermis. When you look at the Pacinian corpuscle, this is the biggest of the mechanoreceptors. Now, this is present deep inside the dermis. And if you notice, the Pacinian corpuscles are the biggest and also the most extensive of the mechanoreceptors. The Pacinian corpuscles are present throughout the palm and help in our sensation of the signals. Now, you can also see that the other type of mechanoreceptors are the Ruffini's endings. The Ruffini's endings are about half the size of the Pacinian corpuscles. They are also present in the dermis. And you can see here the Meissner corpuscle. The Meissner corpuscles are present at the ridges. You can see at these ridges. And the Meissner corpuscles are mainly responsible for the sensation of touch. Then you have the Merkel's discs, which are the free nerve endings. The tip of the Merkel's discs are made up of the non-neuronal cells, which are the Merkel cells. Okay, so all of these cells contribute to the sensation of touch. There is also the hair. We cannot underestimate the role of the hair. Now, the hair are generated by the hair follicle receptors that are present in the dermis. And the hair actually helps in different functions. It helps in the sensation of touch. It helps in thermoregulation. And many times you can see that the change in the conformation of the hair also contributes to many of our sensations like goosebumps. Now, some other types of mechanoreceptors are like the Krause-N bulbs, which basically lie at the border between the dry skin and the mucous membrane, like for example, around the lips, around the genitals, and they look like a ball of strings. Okay, so all of these contribute to our sensation via touch. All these mechanoreceptors, which are in the form of corpuscles, which are also the hair receptors, and all of these are distributed throughout the skin. Now, if you want to understand how the distribution of these mechanoreceptors are on the skin, you can just use a stimulus. For example, you can use a probe, for example, like a pressure probe. And then you can apply this pressure probe at different regions on the palm. And then you can use a recording electrode to measure the frequency of the firing or the frequency of the action potentials that is generated along the median nerves. Now, each of these mechanoreceptors have their own preferred stimulus frequencies. They have their own preferred pressure. So depending upon the extent of the pressure also, some mechanoreceptors are active, others are less active, others are not active. And they also have their unique receptive field sizes. For example, you can see here the receptive field or the area where these mechanoreceptors are distributed varies. Like for example, the Meissner's corpuscles are only present at discrete regions. You can see that over here. Whereas the Pacinian corpuscles are more distributed. Like you can see that you have a big chunk of the palm, a little bit of the fingers, all of these having the Pacinian corpuscle. So the receptive field size is also different. So that helps in understanding the distribution of these mechanoreceptors in the on the skin. Also, the mechanoreceptors have different receptive field sizes. Some of them have small receptive fields, some of them have large receptive fields. Some of them undergo a very rapid adaptation. Some of them are very slow to adapt. Now, if you see here, the Meissner's corpuscles and the Pacinian corpuscles rapidly undergo adaptation. Now, what is this adaptation? Now, for example, if you apply, say, for example, a pressure stimulus. Now, as you apply the pressure stimulus, you see there is a firing, the firing of the axons, right? The action potentials are getting generated over here. Now, after some time, even though the pressure stimulus is still there is no firing here because the system or the mechanoreceptors have adapted. Now, just imagine like you encounter an enormous pressure. An enormous pressure is applied on you. The first time you encounter it, you face the maximum brunt of it. But slowly your body adapts to it. The adaptation is coming because of these mechanoreceptors. Now, some mechanoreceptors are rapid to adapt. Like for example, Meissner's corpuscle, see it's adapting rapidly. You don't see any firing over here. The moment you remove the pressure stimulus, the mechanoreceptors start firing back again. The same goes with Pacinian corpuscles. Whereas the Merkel's disc and Ruffini endings, they adapt slowly. Like you can see here, you apply the pressure stimulus, and then you can see the firing of the axons. But even after the pressure stimulus is there, the firing reduces, but it is still there periodically, right? This is slow adaptation. Similarly, you can see with relation to the Ruffini endings, also an adaptation that is slow, okay? So based on this, you can categorize the mechanoreceptors on their receptive field sizes and also on their ability to adapt. Further, you can categorize these mechanoreceptors based on the frequencies at which they respond. Now, some mechanoreceptors, like Pacinian corpuscles, respond at high frequencies. So just imagine you have a stereo at your home, and then you just put your hand up on the speaker. When you put your hand on the speaker, you feel certain sensations. These are high frequency sensations. These high frequency sensations are coming from the Pacinian corpuscles. On the other hand, if you just move your hand across the speaker, you will see these indentations on the speaker, right? And those, as you keep moving your hand, you also sense a type of touch-based sound. That coarse indentations will create a type of response, and this response is mediated by the Meissner's corpuscles. So on one hand, you have high frequency responses, high frequency touch-based responses that are coming from the Pacinian corpuscles. You have the low frequency touch-based responses coming from the Meissner's corpuscles. So that means you can again categorize the mechanoreceptors based on the frequency sensitivity of them. Okay. Now, once the mechanoreceptors are responding to the touch stimulus, the next thing that needs to happen is for that sensation to be carried to the next regions of the nervous system. And this is done by the ion channels. Like some ion channels based on touch are very sensitive to touch, and the touch can result in the stretching of these ion channels. The stretching of these ion channels will open them, and the opening of these ion channels will depolarize the neurons because of the entry of sodium or the entry of calcium. Some other ion channels are dependent upon certain extracellular matrix proteins. Now, based on the type of touch, based on the type of pressure, there may be a deformation in the extracellular matrix proteins, and these deformations will lead to the stretching of these ion channels, resulting in their opening and the entry of the ions like sodium and calcium, and as a result, depolarization of the neuron. Some others can also open in response to deformations or modulations in the cytoskeletal proteins. Now, these cytoskeletal proteins can deform based on the touch, based on pressure. And accordingly, they can open the ion channels, releasing or contributing to the entry of sodium or calcium, and the positive charge coming inside depolarizes the neurons and carries the signal further forward. Now, one important thing to understand in our body is the ability of our body to be able to discriminate touch. Some regions on our body are more sensitive than other regions. Like, for example, the index finger can be able to discriminate 2 points that are as close as this. So just imagine there are 2 points on either end. If the distance between those 2 points is as small as this that you see here, the index finger is still able to tell them as 2 points. But for example, for an abdomen to be able to tell that there are 2 points, they need to be separated by about 42 millimeters. You can understand how sensitive the index finger is. Just 2 points that are separated by such a small distance, our index finger is still able to make out that they are 2 points. On the other hand, for an abdomen to be able to make out that there are 2 points, they need to be separated by as much as 42 millimeters. Now, the same kind of sensitivity varies all across our body. Like, for example, the big toe, the sole, the forearm, the thumb, all of them have different sensitivities. But the maximum sensitivity is seen with the index finger. Now, you can see here, the dots of a braille are about 1 millimeter in height and 2.5 millimeters apart. Up to 6 dots, they make a letter. An experienced braille reader, basically a reader who depends on touch, can read across a page about 600 letters per minute. It is as quick as somebody who can read aloud. So you can understand how much sensitive our index finger is. Now, why is our index finger so sensitive? It is basically because of a very high concentration of mechanoreceptors. Also, a large amount of the brain tissue is devoted to inputs that are coming from our index finger. And that is why in a 2-point discrimination test on our body, the index finger scores the best. Now, when you look at what happens when we receive a sensory input in the form of touch. Now, here you have our sensory receptor. This can be any of our mechanoreceptors. Now, you can see the sensory neurons are distributed all across the body. Now, you can see here, this is a sensory receptor. Now, the sensory receptor, what you are seeing here is the sensory receptor is receiving the touch stimulus. The sensory receptor is receiving the touch stimulus, and as a result, the touch stimulus is carried by the dorsal root. Okay, it is carried by the dorsal root ganglia, and from the dorsal root ganglia, the nerves get into the dorsal horn of the spinal cord. Okay, so this is what you see here. The dorsal root ganglia carries the impulses from the sensory receptors. Now, this is the cell body of the dorsal root ganglia, and you can see here it is going all the way to the dorsal horn. Dorsal horn is basically the gray matter of the spinal cord. Now, you know very well the spinal cord has the butterfly-shaped gray matter, and then there is a white matter which is surrounding this gray matter. Okay, now this is how the sensory inputs are going from the sensory receptor all the way to the dorsal part of the ganglia. Okay, now it's important to understand the afferent axons are again divided into different categories. Okay, how are they divided into different categories? They are divided into different categories based on their diameter. Now, diameter of the axon plays a very important role in classifying these afferent axons. Now, for example, you have the A alpha, A beta, A delta, and C, or they have been named otherwise as 1, 2, 3, 4. Now, A alpha or group 1 has the biggest diameter, and also it carries the fastest. The impulses are carried the fastest. Group 4 or C has the smallest diameter. That means the impulses are carried the slowest. So basically, the diameter of the axon plays a very important role in how quickly the impulses are carried to the next stage and from there onwards to the brain. Okay. Now you can see here, this is the sensory organization of the spinal cord. Now, this is your sensory receptor, basically any of our mechanoreceptors. Now, as soon as there is a touch that is applied, as soon as some pressure is applied, the sensory receptor is able to sense that pressure, and that signal is then carried forward via the dorsal root ganglia, via the dorsal nerves, and it gets to the dorsal horn. Okay, it gets to the dorsal horn. Now, from here, if you notice this further, what happens? For example, you have the A beta axon. The A beta axon has one of the biggest diameters. Now, it is carrying the impulse. It is coming all the way to the dorsal horn. Now, you see what is happening here? As soon as it is coming to the dorsal horn, it is actually branching out into two. One of these branches is going to the brain. The other branch is actually synapsing in the dorsal horn and is synapsing with a second set of neurons, which are the second order sensory neurons. Now, why is this happening? This branching is very critical for the reflex actions. So remember, sometimes we experience when we touch anything that is hot, or somebody pinches us and then immediately we respond to that, or some bee stings us, we immediately respond to that region. That response is coming because of a reflex action. The reflex action does not need to go to the brain. Here, this particular branching that happens with the A beta axon that is receiving this sensory inputs. This branching is actually dividing the sensory responses to a reflex action, and the reflex action is carried out by the ventral horns. Whereas the other branch is going to the brain. Now this is important for carrying out all the complex set of outputs. Okay, so you need to clearly understand this branching mechanism is a very critical aspect of our touch response. Now, how does the neurons that go to the brain, how do they really connect to the brain? The most important pathway is the dorsal column medial lemniscal pathway. Now let's see how this works. Now here you have the sensory receptors like we saw in the previous slide. The sensory receptors are going via the dorsal horn, and as they go to the dorsal horn, it branches out into two. One is responsible for the reflex action, whereas the other branch is going towards the brain. And before going to the brain, it first synapses at a region close to the medulla, and this region is called as the dorsal column nuclei. Here, a second set of neurons arise, which descend downward into the medial lemniscus. And as they descend downward, they actually decussate. You see, the sensory inputs are coming like this, going in a straight line, but the moment they get into the medial lemniscus, they actually go in the opposite direction. They are descending and then they are going into the opposite direction. This is contralateral decussation. And then from this direction, they traverse to the thalamus. Specifically, they traverse at the ventro-posterior nucleus of the thalamus. They synapse over here, and then a third set of neurons arise, which carry the signal further to the somatosensory cortex for a complex set of responses to be given out. Okay, so that's important to understand over here. Now, what we saw in the previous slide was responses that are coming with the involvement of the spinal cord. There is also another pathway that contributes to the touch responses that arise from our head, the touch responses that arise from our face, and this pathway is called as the trigeminal touch pathway. The trigeminal touch pathway is mainly responding to the touch sensations from the skin that is around the ears, around the nasal areas, around the pharynx, and it is provided by cranial nerves such as the facial nerves, the glossopharyngeal nerves, and the vagus nerve. Now, the pathway that it follows is very similar to the previous medial lemniscus pathway. Now, you can see here, these are the large mechanoreceptor axons that, again, are responding to touch based on the sensory receptors present on the face. These signal all the way to the first set of synapses, and this first set of synapses are happening at the primary sensory trigeminal nucleus. From here, the decussation happens, basically a contralateral movement. And the contralateral movement leads to a second set of neurons that go all the way to the thalamus. They synapse at a region known as the ventro-posterior nucleus of the thalamus. And from there, another set of neurons carry forward the signal all the way to the somatosensory cortex. So understand that there are two pathways, one responding to the touch signals where the spinal cord is involved, called as the medial lemniscus pathway, and another that is responding to touch signals arising from the face, arising from the head, basically involving the cranial nerves, which is the trigeminal touch pathway. Finally, if you go and look at the somatosensory cortex, there is a very unique organization of the somatosensory cortex. Now, the somatosensory cortex is organized according to the responses it receives from different parts of our body. Like, for example, this particular region of the somatosensory cortex is critical for responses of the genitals. The next region to it, the responses from the toes. The touch responses from the foot is the following region, and so on. So you can see different regions of our body have been nicely categorized and classified and separated in the somatosensory cortex. Some regions have a lot of gray matter. Like for example, the touch responses coming from our hands, especially from our fingers, have a lot of gray matter in the brain. The touch responses coming from the lips have a lot of gray matter in the brain. Whereas the touch responses coming from the abdomen has comparatively less gray matter in the brain. Now, all of this have been organized into a map-like structure, and that map is called as the homunculus. Homunculus is basically, in Latin, it means a little man. Now, what is the homunculus? It almost looks like a caricature. Now, when you look at it, the homunculus has been arranged based on the gray matter that has been devoted to the region in the brain. Now, for example, you can see the hands are quite big. That means a lot of gray matter has been devoted to these hands with regards to the somatosensory cortex. The gray matter, similarly, the lips are quite big. That means a lot of gray matter has been devoted to the touch responses or the somatosensations happening from the lip here in the brain. Whereas for the abdomen, the amount of gray matter devoted is comparatively less. So this is a way to represent the gray matter that is devoted in the somatosensory cortex in the form of a caricature, which is nothing but called as the homunculus. So our touch is a very, very unique response that we have. In fact, we can very well say that even without any kind of other sensations, like for example, vision or audition, we are able to identify things by touch. Now, imagine something crawling on your body. You don't need to take a look at it to be able to identify it. Just by understanding these somatic sensations coming from touch, you are able to identify where exactly that particular insect is crawling on your body. Now, all of this is dependent upon the touch responses coming from our mechanoreceptors, and these are some of the most critical type of somatic sensations that are present in our body. So I thank you.